Right, perfect. All righty, welcome everybody. Thanks for coming out for this month's edition of Pizza and Programming. This month we have Thomas Stricker, who's an assistant professor in Pathology, Microbiology, and Immunology.
Yep.
PMI, did I get that right?
Yep.
And also director at...
Associate Director of Vantage.
Of Vantage.
Yep.
And he's going to be walking us through nuts and bolts of next generation sequencing analysis at ACCRE.
Yep. Thanks, Will. So I've never given a talk quite like this before, so we'll see how it goes. We're gonna. I wasn't exactly sure on the audience, so we're gonna start with a little bit of sort of how Illumina sequencing works and the molecular biology behind it, because that is going to be important for the analysis. We'll define some terms, and then we'll sort of walk through sort of how I do things for a couple different pipelines. And we'll sort of stop there. So first human genome took 10 years and $2.7 billion to sequence. We can now sequence one for $500 to $1,000 in a few days. So this is a massive change in our ability to generate a huge number of bases. This decrease here and then this big drop here are both almost entirely due to flow cell sequencing by synthesis, as essentially created by a company named Illumina. Well, created by a company named Solexa, but by Illumina. All right. So just in case we have some non-biologists in the crowd, there are, so DNA is a double-stranded polymer of 4 bases, A's, T's, C's, and G's. And the order, or the sequence of those, are what we call the genetic code. A always pairs with T, and C always pairs with G. And about 40 years ago, Edwin Sanger and colleagues invented what is called Sanger sequencing. It was then modified by a number of people, including Lee Hood. And the way that we sequenced the human genome was we had a tube with a single DNA molecule in it. We made multiple copies of that DNA molecule. And then using a polymerase, and then using primers and polymerase, we had a mixture of regular DNTPs and what we call terminated DNTPs that were also labeled. And by terminated, no other base could be added onto them, right? So when that small fraction of terminated bases were added to the polymer stopped. And as we made lots of copies of this single molecule, we would get a ladder. And then we could separate those either on a gel or by capillary electrophoresis. And using a laser, we could scan to see which base came next. And that was how we were able to determine sequence, right? So, like I said, using this on a human genome cost about $3 billion. That's not tenable if we want to sequence lots of genomes. So what happened was 3 things. First, we had that original draft of the human genome. So if we didn't have a draft of the human genome, none of this is really possible. Second is in vitro amplification or cloning. So we can take, say, the entire human genome, which is 3 billion base pairs, chop it up in tiny bits, and add universal adapters onto the end. And so inside a tube, we have copies of every piece of DNA in the human genome, rather than having one particular piece of DNA per tube. And the third is flow cell sequencing by synthesis, and we'll talk about that in a minute. But essentially, this is a picture of an old flow cell from an old Illumina sequencing machine. So each of these is a lane. Each of these lanes is coated with oligos, and as we flow our library with our universal adapters over the end, pieces of the library stick down, and so we can sequence billions of molecules all at once, rather than sequencing one molecule in a tube at a time. Yeah, so and the way this works is we have our DNA, our library, molecules from our library stuck to this flow cell. We're going to add we're going to add all 4 bases, A, T, C's, and G's. They're all terminated, and they're all fluorescently labeled. So we add 1 base to every molecule on that flow cell, and we take a picture. And then we reverse the termination and remove those fluorescent labels so that we can add the next base. We add the next base, and we take a picture, right? And you stack those images up, and you're able to sequence 100 to 150 bases per each of these dots times millions of dots. That gives you terabytes of data. You can see, we're going to come back to this, it's, each of these spots are different colors, but some of them are different shape. You may not be able to see in the back, but there's a white one here. So there's different quality of the sequence. And so we're able to infer how confident we are in the base call based on shape and color characteristics. So I said each of these is a molecule, and that's our DNA molecule, and that's kind of true. Each of these is many copies of the same DNA molecule, and that helps the fluorescence is brighter. But we can have what's called phasing, where some but not all of the molecules get their terminator reversed, and then the color gets to shift, and you start to lose your ability to call bases. And so the quality of the sequence degrades over time, and it does so variably. All right, so I'm going to use some terminology, so I just thought I would define it. So single end or paired end. So single end means we're going to sequence one end of our DNA molecule. Paired end means we're going to sequence both ends of our library. And there are reasons for this. And then we'll talk, you might hear people say, I probably won't use it here, 50 base pair or like 75 paired end. So that means we're sequencing 75 base pairs in from each end. Or 150 base pairs, we're sequencing 150 base pairs in from each end. So a lane means we have, so this is a flow cell from an Illumina 2500. So there are 8 lanes to this flow cell. And we can load a pool of libraries onto each particular lane. And we can load different pools onto each lane. Or we can load the same pool across all 8 lanes. But we need to pay attention to which lane our sequence came from and when we recalibrate quality scores, which I'll talk about later. Barcodes are indices. So especially these days, we generate more sequence per lane than you generally need per library. So we actually pool libraries together and sequence them all on the same lane. And then in order to tell them apart, the universal adapters we add have unique barcodes in them that are assigned to that library. And so then we get all the sequence from the lane, and then we assign each individual sequence to its particular library based on the barcodes and the adapters that we use. Library is the base unit of preparation. You give me DNA or RNA, and I make a library out of it. And sample is a single individual. So you might make multiple libraries from the same individual. You'd want to give them the same sample name, but then have a unique library name for them. All right, file names that we will use. So FASTA or FASTA, this is the raw sequence file, or not raw sequence file, it's a sequence file, A, T, C's, and G's. So reference genomes, reference transcriptomes, etc. will be as FASTA files. FASTQ files are what we produce in Vantage and hand out to folks for analysis. So this is sort of our base unit of analysis. It is a FASTA file plus quality scores, and I'll show you what that means in a minute. A BAM file, so that's an alignment file. It's in binary format to shrink it down. So after we have aligned our FASTQ file to genome, what we have produced is a BAM file. A VCF file is a variant call file. So when we go to genotype, the output of a genotyping run is a variant call file. Indexes, so when we talk about indexes for libraries, we mean the barcodes. When we talk about indexes for analysis, these are essentially lookup files or lookup tables that you can generate for FASTA, BAMs, and VCFs that make access to these files very quick for the computer program. And a lot of tools that we use require an index file to have been generated in order for the tool to run. So sometimes you'll go to run a tool and it'll throw an error, and the error will say there's no index file found, so you just need to generate an index file. MAF is a mutation annotation file. So this is often an annotation of VCF that includes information about the variant. Has it been seen previously in the human genome or in the human population, et cetera? I'll talk about annotation at the end. A GTF or GFF file provide information about gene structure. This is particularly important for RNA-seq analysis, but essentially the start and stops of exons and start and stop of coding sequence and start and stop of genes. Interval list. So let's talk about BED first. So this is a BED file. So a BED file is essentially just a set of genomic ranges. So chromosome, start, and stop. So start position, stop position on that chromosome. BED files come in many varieties, so you can have a whole bunch more information about these intervals out here, like maybe which gene it's in and which strand it's on, or you're interested in that sort of information. But this is a base BED file. And interval files are BED file, because we needed another format to have the same information. Interval files are basically BED files, but they're required for Picard, which we use a lot in our genotyping pipeline. And you can generate those from BED files using Picard BED to interval file. So we have multiple files sometimes that have essentially the same information. All right. If we're going to sequence the human genome, I think there's some important things to know about it, and also just an idea of like genome size for other model species. Human genome's about 3 billion base pairs big, long, however you want to say it. About 1 to 2% of that's coding. The rest of it is not. Something like 5 to 10% shows some evidence of positive selection, depending on which paper you read. So lots of the human genome is basically junk. And so we're going to develop techniques to deal with that. So one of the things we might be interested in doing is just sequencing regions of interest rather than sequencing the whole human genome, because sequencing 3 billion base pairs takes a lot of money still. And so one method for target enrichment is hybrid capture. This essentially starts with, so you take your DNA, you chop it up into tiny pieces. You hybridize those pieces to baits, which are produced by a company that are homologous to the regions of interest, maybe every exon in the human genome. Those baits have biotin on them, and then you can use streptavidin-linked magnetic beads to pull down your DNA region of interest, wash away everything else, and then just sequence your exons. This works pretty well. We're getting better and better at it. It has an incredibly large capsule space, anywhere between 30 and 70 megabases, so 70 million bases out of the 3 billion. It allows you to analyze things, not just point mutations, but copy number and translocations. Variants outside the baits might be sampled. So it doesn't have to be a perfect match. You get sort of a splash around where your bait sits. When you pull things down. It does require a reasonable amount of DNA. We're probably lower than 250 nanograms at this point. The sequencing is a little inefficient, partly because we don't wash away all the bad DNA that we're not interested in, and partly because of the splash where we're not sequencing just over the baits exactly. That means we have to generate more bases than you would think in order to adequately cover what we want to cover. There's also amplicon-based target enrichment. So that is essentially we put PCR primers at regions of interest and we just amplify those pieces. You can go to really small amounts of DNA for this, which is pretty handy. The PCR can be uneven because we're doing multiplex PCR of maybe thousands in a single tube. And the number of genes is limited. You really can't go much above 50, and even then you're not doing full gene sequencing. It's often just pieces of those. But it does enable you, it's incredibly the sequencing then is almost all on target. So it makes sequencing incredibly cost effective. And so you can get very deep for these regions. And by deep, I mean have lots and lots of reads that cover those areas. RNA-seq. is a very common thing that people do in Vantage. It's probably one of the more common assays we do. And that is essentially we purify RNA. We convert it to cDNA. We build sequencing libraries out of it, and then we sequence the RNA. There's a couple things to consider here. So 99-ish% of RNA in a cell is ribosomal RNA. We don't want to sequence ribosomal RNA because that's boring. So we need to purify messenger RNA. There's two ways to do that. You can use poly-A, so that will give you only messenger RNA. Or you can use ribo-, which actually removes. Poly-A selects for adenylated messenger RNA transcripts. Or you can use ribo-, which will remove the ribosomal RNA and leave everything else, which includes both adenylated and non-adenylated non-ribosomal transcripts. So things like long non-coding RNAs, some of which are not adenylated, would be available in the ribo-, but not in the poly-A. But only things generally over 200 base pairs or more. If you want to sequence small things like microRNAs, you need to use a special purification and library build protocol. So you have to think about what you're doing before you get to RNA. But RNA gives you a lot of information. You can essentially count the reads and ask how many, count reads for each gene and say how much of each gene is expressed. You can look at isoform-specific quantification. You can identify new genes and isoforms. You can identify fusion genes where two genes have been translocated to each other. And you can genotype off of this as well, which allows for things like looking at allele-specific expression. I'm not going to talk about ChIP-seq analysis today just because I don't have time, but it is probably the other most common thing that we do. And very briefly, we take DNA, we cross-link the protein to DNA, we sonicate the DNA to break it up tiny pieces. We use an antibody against our favorite transcription factor to pull down the DNA to which it is bound. We wash everything else away. And then we uncross-link and sequence the DNA to identify binding sites of transcription factors, histone marks, et cetera. A lot of the sort of QC alignment and then counting that we do for RNA is, in the end, kind of similar for ChIP-seq as well. Oops, did that already. All right, genome versions and gene versions. So there's more than one version of the human genome because we try and improve them as we go along. There's two in most common use are HG19, also known as GRCH37, which came out, I think, in 2009, and then GRCH38, which came out in 2013. So this is the most recent version, and unfortunately, almost no one uses it. The reason no one uses it is because a lot of our annotation for things that we want to do, so we have, and we'll talk about this later, in large catalogs of human variation, whether they've been seen before in the human population or not, are on 19, and they're not on 38. So if I want to say, if I find a variant, I want to know, is it a common polymorphism that I've seen in the human population or not? It's going to be much easier for me to look to see if it's in 19 than to see if I've aligned to 38. And that's because the coordinates are different, which I'll show you in a minute. If you're doing RNA-seq, you're going to need to decide what set of gene models you use. So we don't agree on what constitutes a gene, basically. So there are multiple, these are just a short list. So there's RefSeq, GenBank, and Ensemble, as well as others. RefSeq is the most curated. So we're pretty confident that all those genes actually exist in nature, but we do not, but it is also then the shortest. So there are probably real things that exist that aren't in RefSeq. Ensemble is probably, I would say, the loosest, and then GenBank is sort of somewhere in between. So this is P53. This is on HG19. These are RefSeq isoforms for 53. These are GenBank isoforms for 53, and these are ensemble isoforms for 53. And so they don't agree, right? There are isoforms that exist here that don't exist here and don't exist here and vice versa. And if you use, say for instance, if you use this, if you use ensemble, then reads that map to locations that aren't present in genes in RefSeq will be counted as P53 expression when you collapse to a single gene model. There is no right answer to this. You just have to pick what you are, it's based on your experiment, your depth of sequencing, and what you are interested in. And then, so I can't see, but so these are the coordinates for 53 in 19. And when you look at 38, they're different, right? So if I tell you that there's a variant at in P53 at base pair of 7.5 million or whatever, and you go to look at 38, but I'm at 19, you won't even be in 53 anymore. So you really need to know what version of the genome you're using, and a lot of times it matters for, or what version of the genome someone else has used. Also, the isoforms for 53 have changed in between 19 and 38. So, all right. FASTQ files. So this is what we produce in Vantage and send to you. This is an example of a paired end read, or paired ends experiment. So same sample name, and then read 1 and read 2. So these are paired. If single end read, they would not have R1 or R2, and there would just be a single FASTQ file for that. This is what the head of a FASTQ file looks like. So we have a read name. There's a bunch of information in that read name that we'll actually extract later. ATCs and Gs, and then these are ASCII characters that are the quality score. So they're Phred scaled, and it's a negative log 10 of the empirical error rate as determined by Illumina. And so each of these is assigned, each of these ASCII characters is assigned to a number. Unfortunately, the encoding for error scores has changed over time. It's been pretty consistent for a long time now, so anything that we produce now is definitely 1.8, which is Sanger, so it's Phred plus 33. However, so if any of you are primary analysts and you are getting data for which is unknown provenance, or you're getting old data, or you're downloading data from an outside source and you don't know when it was sequenced, you ought to check. There's a script. I have guessencoding.py. There's other scripts you can get that will tell you what the Phred scaled encoding is for your quality scores before you do anything. Because if you don't know what it is, you can't inform the aligner and other programs what quality score metrics you're using. All right, so when I get sequencing data, the first thing I do is QC the raw sequencing data with a program called FastQC. I'm then going to trim to remove adapters. So sometimes our inserts are really short, and we end up sequencing into the adapter on the other side. And we want to cut those off. And also, there's no reason to try and align low-quality sequence, so especially if it's a bunch of ends. So we're going to they couldn't even call it base, so we're going to trim those off as well. I'll re-QC that clip data just to take a look. And then I'll use a program called MultiQC to summarize the clipped, or summarize my FastQC files. The MultiQC, I just run in a simple bash script. It doesn't require really scripting, just find all the files. I store all the FastQC files in a particular place. So, let's see, all right. All right. So most of my early scripts were written in Perl because they're like 5 years old, but I just use them over and over again. So when I'm given a project, and this is where ACCRE comes in super handy, I am given, I'm not given 2 sequencing files. I'm usually given dozens, right? And so I want to analyze them all the same way pretty much at the same time. And so what I do is I have a Perl script that, and I tell it the path where all those sequencing files are hidden at command line. And then it's going to, I set some variables for where I want to print things out. And I find all my FASTQ files are gzip, so I find all those files. And then I essentially generate a bunch of batch submission scripts and send them to the scheduler automatically. So that it's doing the same thing for every file and is essentially running FastQC on all of them. The output of a, let's see. This is an output of a FastQC for a single sample. And first thing we get, so there's a bunch of metrics here. So first thing we get is the binned quality score by base for sequencing. So this is 75 base pairs. And the quality score is very high across the whole length of the read on average. Right? Old sequencers, you often see a decrease as over time due to this phasing. As the enzymes have gotten better and better with Illumina, you see less and less of that now. But that's also why we'll sometimes trim off the back end. And then there's a bunch of other information here that can be very useful. So this is, I think, quality, I can actually look here. Per sequence quality scores. All right, so this is a failure because, so this is, each of these are ATCs and Gs, we'd expect about 25%. There's a decided non-randomness to this end, to the 5' end. This is RNA sequencing, and so even though it says it fails, it didn't. This is what you expect for RNA. So we random prime our RNA, and random primers are not random. And so we have sequence bias at the 5' end that then eventually smooths out. So you have to be careful with QC, right? So not everything that says it's a failure is a failure. You have to know what you're sequencing. But lots of other information here that can be very useful in interpreting your results. And then you can pull this. Is it this one? Yeah. So you can pull. So I generate one of those files for every FASTQ file for which I have 2 for every sample. So if I have a large sequencing project, that's a lot of HTML files to go through. Multi-QC pulls them all together for me. So I can look and sort of rapidly identify. So we have some bad samples here with a high duplication rate. So those are probably, these are RNA from FFPE. There's probably a very small amount of RNA got put in, and so we had a lot of PCR duplicates. And so we can, this is helpful in terms of being able to diagnose problems, throw out bad samples, decide whether we're going to resequence or not. And you can go through this HTML doc much more quickly and sort of look at the look overall at the project. So we'll go back this way. All right. So very quickly, we'll run through an RNA-seq expression analysis. We're going to start with a splice-aware alignment. So the average, so first of all, human genes are not in one piece, but they're actually broken up into tiny pieces. And then those tiny pieces are stitched back together during a process called splicing. The median exon size in the human genome is about 140 base pairs. The median intron size, so the exon is what we call the coding region, right? The intron is the non-coding region that split up the exons. The median size there is something like a kilobase, right? So we've got a problem where we have to place 100 base pair fragments in a 3 billion base pair genome. And the places where they need to align to are small, 140 base pairs. And the places, and some of those reads may cross from one exon to another. And those islands might be far, as much as 1,000 base pairs away from each other or more. So there are a variety of aligners that have been developed to deal with this problem. The three most common are Top Hat, Star. I would say the three most common anyways are Top Hat 2, Star, and RCM. I have migrated to Star these days, but your mileage may vary. So we're going to align with our favorite aligner that's going to produce a BAM file, our binary alignment file. I'm going to run Picard and RSC-QC for alignment QC on all my BAM files. I'm going to pull all that together with multi-QC again. Then we're going to assign reads to transcripts to produce count files. There's, again, a variety of ways to do this. HitSeq, feature counts, cufflinks. There's others as well. RSEM will actually do it. And so what that generates then for each sample, for each gene, I have a number of reads that cover that gene. I collect that data into a matrix and then read that. For differential expression, you can read that into R. There's a multitude of packages that you can use for differential expression. DSeq2, Lemma, or LemmaVoom, and EdgeR are probably the most common, but there's certainly others as well. All right. All right. So same idea. All my FASTQ files are in a folder, right? So I'm going to run my program and tell it which folder that is. And this makes my code a little bit portable, so I can just, if I get a new project, I just have all those sequencing files in a different folder. Just run the same script on that folder instead. There are some changes that you might have to make. So what's commented out here are the human genome and the index that I built for the human genome. And so what's running here is mouse. So this was for our mouse project. And we're specifying a FASTA reference. We're specifying an index that has been generated. So for fast lookup of that index, or that reference. And then we're specifying gene models in the GTF format. So then this is paired end experiment. So I have a little piece of code here where I'm going to find all of my sequencing files. I'm going to go through each of those sequencing files and make a hash of hashes where the first key is my sample name extracted from the file. And the second key is whether it's read 1 or read 2, provided it matches the two ways right now that Vantage encodes reads. If they change the way they encode reads, I have to fix my code, and I really hate it when they do that, but they do it every once in a while, and you just have to deal with it. So having built that hash of hashes, I'll then step through, write an output script that I can submit to the scheduler for each sample. So go step through for each key. The first key, remember, is sample. So for each sample, I can then print out the star command, and here we're specifying read 1 and read 2. And so it generates the command I need with all the stuff I need, and we'll just run it. So I can run the same command on all the files, and it just takes a few minutes to run this. I'm sorry I'm still using set packages, Will. I have been switching to LMOD for some things. I just haven't got all my code switched over yet. So as I get there, we'll have some scripts later where I do have switched to LMOD. All right. So BAM files are one of these days I'll remember which direction to go. BAM files, there's a ton of information in a BAM file. We can't look at a BAM file. Let me see if I can. We can't look at a BAM file natively because they're binary. So we have to use a program called samtools and then a command called view in order to look at this BAM file. This right now we're just looking at the header. So the header tells me, it actually tells me what genome I aligned to. So there's a chromosome and length for each of those. And then it actually tells me what commands were run on that genome. So STAR does a 2-pass alignment, or can do a 2-pass alignment. So that's what I do. So it aligns to the genome using known junctions, but also then identifies novel junctions as it aligns, and it realigns again using those novel junctions plus known junctions. And that's one of the reasons I like STAR. It's particularly good for genotyping RNA. It reduces false positives. So that flag H gives us the header. So if you pull that down, then I have to... so it's millions and millions of lines long. So that's why I killed it. But this is what that file actually is, which is an alignment. So it tells you, so this is the read name. This is what we call a cigar score. And it's a bitwise flag, and it tells you information about how the read was aligned, how its pair was aligned, if they're in the proper orientation, and a bunch of other information. This aligned to chromosome 1. The read starts at this base. So there were 61 bases skipped and 14 matched. This is not a great read. There's some other flags that are sent. This is the actual read. These are the quality scores. And so there's a lot of information in this BAM file. And let's see, where were we? And then, so the summarized metric QC is here. And so after having generated all the BAM files, I'll run a couple different QC programs on my on each of the alignment files, and this pulls all that information together and provides a tremendous amount of useful information. The percent of bases aligned, the number of reads aligned, the percent duplicate reads, the percent GC, so that's all here for each of those libraries. This gives you your gene body coverage, so we don't see a tremendous 3' to 5' bias, so even though this is FPE, we don't have a lot of gene degradation. So if we had a really bad, so if your mRNA is highly degraded, you might just see a lot of sequence at 3' and not a lot of sequence at 5'. This gives us insert distance. So we have these are some of those libraries that had a high duplication rate. They also have a high primer dimer, so we have a very peaky insert. You can also see that here. These are read duplication levels. This is the most interesting to me, though. So this is junction saturation. So this gives you an idea of how well you have saturated both known and unknown junctions, so splice junctions. And to me, for RNA-seq, it gives you a really good idea of how much additional sequencing will help. So, and we can just look at, so these are known junctions, and then these are novel junctions. And so we're saturating, for most of these libraries, we're saturating our known junctions. So we've probably generated enough reads. For novel junctions, we aren't, probably for lots of them, we probably aren't saturating, but we are seeing the more reads we generate, the less payoff we're getting. So if we're really interested in novel, maybe we would sequence some of these libraries more. This is FFPE, so this may not be the best place to go looking for novel junctions, but I find that to be pretty helpful in assessing whether we want to need additional depth or sequence on a library. And then we'll run, given those BAM files, we'll then translate those to count tables, basically. And these are not certainly the most interesting files in the world, but so oops, kill. Sorry, I should have used head, not cat. So gene, each of these is a sample. Each of these is a number of reads for this gene in each sample. And this is the information then that we can use in like a negative binomial model to determine differentially expressed genes between our two subtypes or whatever experiment we're running. So at that point, I'll jump to R, and usually on my desktop, unless I'm using an incredibly big data set with like thousands of samples, R at my desktop is sufficient for that. All right. Okay, I'll try and go through this relatively quickly. So genotyping. This is the best practices from GATK, or Genome Analysis Toolkit, which is written at the Broad. Their webpage will be up later. Please go there. I'm just going to go through these steps pretty quickly, but there's a tremendous amount of information there. And their website and their tutorials are incredibly helpful, and I can't recommend them enough. I'm also not really going to talk about how most of this works. I'm just going to talk about how we do it. I don't have time to talk about it. It would take like a half-day workshop to talk about how all of these work, right? So, but what we're going to do is we're going to align to genome. So this is DNA. We're going to align to genome. We're going to mark duplicates. So things that are obvious PCR duplicates, we don't want to count because that would be like counting the mutation twice. We're going to, those base quality scores, those are, that we use, those are inaccurate, so we're going to recalibrate those. We're going to, for each individual sample, we're going to call genotypes, and then we're going to combine those files into joint genotyping, and then we're going to filter those variants either with a either hard filters or actually building a model based on known variation and filtering for our using that model. So this is IGV. IGV is Integrated Genome Viewer, runs in Java. You can download it from the Broad as well. It allows you to visualize BAM files. So these are individual reads that are piled up at a particular site. And this is a place where we have called a non-reference heterozygote. So you can see about half the reads have an A, where A would be the non-reference base. IGV is very useful for visualizing. You can visualize all kinds of stuff there to take a look at BAM files, WIG files, that sort of thing. All right, so this isn't in best practices, but the first thing we need to do is add read groups. So read groups are a set of read generated from a single sequencing run. So usually you will have a sample, you turn that sample into a library, you send that library to Vantage. We generate a single sequencing file off a sequencing run, and so all your regroup information is the same within your FASTQ file. But you can imagine situations where you didn't get enough sequence, so we sequence it again, or you needed lots of sequence, so we split it across lots of lanes, or whatever. So you may have, some of these may change, so you're going to end up merging those files together. And we need to keep track of which read came from which run. And there are a few pieces to read groups. So ID is our flow cell ID plus lane. I'll show you what that means in a minute. So every flow cell has a barcode on it. And remember, we had those lanes 1 through 8. So this is going to tell us that read came from this flow cell barcode plus lane. The platform unit is the flow cell ID plus lane, so ID plus sample ID. And then your sample ID and library ID usually are the same, but not always. So this is back to the FASTQ file. And this right here is the barcode, and this right here is the lane. So I don't have time, we're running out of time, but in my script what I do is I open up I just take the head of each BAM file, and I extract this information, and then I use that to add read groups to every file in Picard. And that way we know which read group each alignment file is associated, or we have assigned read groups to each of our alignment files. Yep. So this is the web page. Software, Broad Institute, GATK, best practices. Also, I do want to definitely mention, like, all this stuff is time sensitive, so what's considered best practices today will not be best practices a year from now. And that's just true. And so, it is what it is. This stuff moves. Okay, so once we have gone through getting our reads ready, adding read groups, marking duplicates, we have our and base recalibration. We have our analysis ready reads. We're going to go to haplotype color or joint color. So I actually do all of these steps in a single script. So they generate intermediate BAM files. So as I generate those files, when I get to the end, I delete those files. So we don't need to keep them. And BAM files are big for exome, a couple gigs. So a couple gigs times a couple hundred samples, and all of a sudden you're blowing up your storage on ACCRE, and you need to get rid of some files. So my script deletes those automatically, which is why I try and generate all of them in a single set of steps. So now we're ready for our joint caller. And so we run haplotype caller on each individual sample. So we're going to feed it a BAM to haplotype caller. And then the output there is a set of SNPs and indels in what we call a GVCF. And then we're going to take our, once all of those are generated, we're going to put them all together in a single table using genotype GVCFs. And so if you have 100 variants, or 100 samples, you'll have 100 of these variants that are in this command. So again, I just write a script that pulls all those together and then generates the sbatch command to send this to send it to the scheduler. This can be slow. This runs very quickly. You can do things like so if you do exome sequencing, you may not want to genotype against the entire genome. You don't expect non-reference bases outside of where you sequenced, so you can limit your you can provide an interval list of just the exons in the human genome to shrink your genotyping space, which will make it go a lot faster. So this will generate what we call a variant call file. Let's do this way. So first of all, at the top end of the variant call file, there's a ton of useful information. So there's going to be a bunch of definitions about what some of these abbreviations are. It's going to tell you how this file was generated, which is always really handy in case someone else is handing you a file. It's going to give you what genome you used for genotyping. And then each line is a variant. So it gives chromosome position, the reference base, the alternative base, the quality of that base, and then a whole bunch of information about that base. So for instance, the allele fraction here is 50%. So it's almost certainly just a heterozygous germline variant. And we'll talk about what some of these other flags mean, but we can use them for genotyping or filtering. All right, so once we've generated, haplotype caller is built to be sensitive. So we're going to call stuff that isn't real, and then we want to filter those out. We have two choices, VQSR, which is a recalibration of our variant quality scores, or hard filtering. So recalibration is recommended for germline human variants where we have 30 or more exomes, or model organisms for which the appropriate training sets exist and you have 30 or more exomes. If it's a non-model organism, or it's not germline, so it's somatic, or you're doing RNA, so it's not DNA, you'll probably need to use hard filters. We'll talk about that in a minute. Recalibration has to be run twice, so you have to split into SNPs and indels. That should say indels, I apologize. So we're going to train a model on a set of variant statistics for variants that we know to be true and exist. And then we're going to use that model to determine the probability that our other variants are true, if that makes sense. So these are the steps. So you're going to run your variant recal, and then you're going to provide it a set of known variants. And these are sort of our test set that we're going to build our model again, and then we're going to test that model on our unknown, our novel variants. Remember how we talked about different genome versions and so on and so forth? You have to make sure that this matches the genome version that you have aligned to. Otherwise, you have gotten all the way down here, and nothing works anymore. And that's a real bummer. Not that that's ever happened to me. And then here, this is the same thing for indels. So we're doing independently for indels and SNPs. And then, okay, so hard filtering, you are going to, and if you're going to hard filter, you're going to set a bunch of parameters for quality metrics. And then anything below those will not get passed through your filter. I'm going to real briefly, we'll talk about the kinds of things you might filter for. So quality by depth. So we have Phred scaled quality scores for every variant. But each read you have contributes a little bit to that. So this normalizes by depth. So things that are sequenced to high depth will have high quality, but they could still be bad. We can have, let's see, what do we have here? So Fisher strands. This is a Phred scaled p-value from a Fisher exact test looking for strand bias. If you have all the variants are only called on the negative strand and no variants are called on the positive strand, that's probably a false positive. We're going to throw that out. And so on. So there's a whole bunch of things we can look at that we can use to hard filter. That said, if you are going to hard filter, please go to these websites and read what actually you are filtering on. And there are recommended sort of starting points for these, but they can change for every project, and you really need to be careful with what you're doing here. Okay. And then, so annotations. So now that we have our variants, we want to know something about them. There are a number of ways to do this, including ANNOVAR, VEP, or others. I use a variant effect predictor from Ensemble the most. These essentially access publicly available data resources. So catalogs of human variation, things like dbSNP, ACCRE or Nomad, exome variant server, etc. So these are, so like ACCRE is 60-some thousand, 68,000 human exomes and variants called from those, aligned to HG19, which is why we still use HG19. Nomad is something like 120,000 exomes. So this lets us know, has this variant ever been seen before in the human population, and how many times? Things that are common are less interesting in general than things that are rare. You have to be careful with these. So there are tumor exomes in here. There are ture variants in here. If you are interested in cancer, you can't just blindly say if it's ever an ACCRE, I'm never going to look at it. You can look at, there are lists of clinical significance, including ClinSig, cancer hotspots, and others. There are measures of conservation or deleteriousness. And so I use these. I produce, so this produces a table. So I take all my annotations and produce a table. It didn't work very well. I must be missing something. So this is actually supposed to be an HTML table that's sortable. So I can actually scroll through and sort and then look. But it provides, this has a ton of information in it, including like exact allele frequency, so how many times these variants have been seen before in the human population. So I produce this for every project that we do, and it makes it sortable. And then I also will often produce these. This is a radar map. So these are 6 measurements of deleteriousness of a particular variant. The red is what would be considered damaging, and the blue is what this variant measurements were under those scores. So this variant would be considered damaging in at least 5 of the 6 measurements. So this is probably likely a damaging variant that would rise to the top of interest of when you're looking for novel mutations or novel causes of a disease. Yep. Yep. So I'm going to wrap up. The only other thing I want to say, so we'll skip genetic genotyping. In the future, there's a bunch of things coming out, including single-cell RNA-seq, and of particular interest, I think, to folks with ACCRE, GATK4 is coming out in January, and that will be Spark-enabled, which will probably change the way we do some things. Yeah. That's interesting. Yep. So I haven't quite wrapped my head around what that quite means for my workflows yet, but we'll get there. So.
We have time for 1, maybe 2 quick questions.
Yeah.
In the early slide, you talk about QC for the past Q5, and you chop the adapter.
Yep.
The bad quality waste.
Yep.
How do you define the bad quality?
So I use a quality score of 10 or less, 7 to 10. Yeah. So.
So in the end or in the beginning of the
So usually, quality scores usually decrease over time. So it's usually the 3' end of a read that you're trimming, and that's also where you're looking for adapters as well. All right.
Jim. Go ahead.
You just now you notice that 2 kind of the QC. One is for weakness, another is for the hard filtering. In order to get a good quality of the genotype quality, can somebody combine both of them together or just separately? Use one kind of method.
So whenever you're filtering, you're always drawing a line between sensitivity and specificity, right? So you shouldn't need to both do recalibration and hard filtering. And recalibration is generally preferred if you can get away with it. But sometimes, either for your data set or you don't have enough samples or whatever, you need to hard filter.
You mean does somebody combine both of them together?
Not usually.
Not
usually.
Just
No.
choose the one.
Just choose one. Yep.
Great. Help me think, Tom.
Wait a few minutes. I'm sure they'll come find me.
Yeah.